Greetings, ladies and gentlemen. My name is Chigwaya, and I'm going to be presenting on a topic titled Browning in Fuji apples explored via X-ray computed tomography and porosity mapping. Fuji apples are prone to browning during long-term storage. Browning incidence is largely influenced by the fruit tissue porosity. However, there is considerable variability in the distribution of porosity within a fruit. Hence, non-destructive methods to map porosity of the entire fruit are indispensable. Fruit tissue porosity is normally higher in the cortex region and lower in the core region of the fruit. The objective of this study was to use X-ray computed tomography and porosity mapping to determine the extent to which different fruit morphological and microstructural properties influence susceptibility to carbon dioxide-induced internal browning. These properties included fruit size, radial and axial porosity profiles, density, and mean porosity. Fuji apples were harvested from the series area during the commercial harvest window in the 2018-2019 growing season. 34 intact fruit were scanned using low-resolution X-ray CT before subjecting the fruit to carbon dioxide stress to determine porosity profiles, fruit size, and porosity. This enabled us to have a grayscale image and a porosity map of each intact fruit before treating the fruit with carbon dioxide. Carbon dioxide stress was achieved by storing the fruit in an environment with 50% carbon dioxide for 3 days at room temperature. After the stress treatment, fruit were evaluated for browning incidence, and cylindrical samples were obtained from the browning affected and unaffected fruit. These samples were scanned with high-resolution X-ray CT to determine porosity. This whole procedure allowed us to determine porosity profiles, porosity distribution within each fruit, tissue density, and fruit size. The browning observed in this study mainly occurred in the core region of the fruit. As shown by the first graph, larger fruit were significantly more prone to browning. Late harvesting of fruit usually results in harvesting of larger fruit that are more prone to browning. Increased oxygen minimal value as fruit size increases heightens the risk of fermentation and browning incidence. The oxygen minimal value refers to the lowest oxygen concentration below which fermentation due to hypoxia is more likely to occur. The second graph shows density and porosity. Fruit that developed browning had a higher density and lower total porosity compared to fruit that did not develop browning. Density is inversely related to porosity, and denser fruit have a low porosity and are more likely to develop browning. Determination of fruit density at harvest may be a reliable indicator of fruit susceptibility to browning during long-term storage. There was a significant decrease in affected tissue porosity after the carbon dioxide stress treatment in fruit that developed browning. This is likely due to membrane damage and loss of cellular compartmentalization caused by a shift from respiration to fermentation. Membrane damage causes leakage of cellular fluids into the intercellular spaces, and this reduces the porosity of browning affected tissue. Flooding of the intercellular spaces and reduced porosity represents the initial stages of carbon dioxide-induced browning before the cellular fluids diffuse out of the fruit, leaving cavities within the fruit tissue. Porosity maps generated for Fuji apples in this study showed the distribution of porosity in the entire fruit. Porosity was low in the core region and high in the cortex region, meaning that the core region is at a higher risk of developing browning due to its low porosity. Size of the fruit significantly influenced browning incidence, with larger fruit being more prone to internal browning. It is important to avoid longer storage durations for larger fruit, as they will be at a higher risk of developing browning. To avoid the production of excessively large fruit, it is important to carefully control pre-harvest factors such as crop load, canopy position, nutrition, irrigation, and harvest maturity, which influence the final fruit size. Further studies into possible correlations between density and browning incidence may help develop a density-dependent predictive model for browning incidence. I thank you.